All right, everyone. So it's 10 a.m. Eastern, so we'll go ahead and get started. Welcome to the second day of our Heart MPS webinar. I'm glad to have you all here, and I'll give a quick introduction. So I'll just read this disclaimer right off the slide. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including cost, price, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. Okay, so I'll give a quick introduction to the 3Rs Collaborative and this webinar series. So we have a Microphysiological Systems initiative, which seeks to increase adoption and regulatory use of MPS. We have over 49 member institutions and over 100 members, and you can see all their logos here. Our initiative aims to provide thought leadership and expert consensus from developers combined with other stakeholders, facilitate appropriate discussion, collaboration, and sharing between commercial developers, create direct engagement between developers and regulatory agencies on the use of MPS, develop external partnerships and collaborations to provide perspectives from developers throughout the field, and create resources to facilitate increased engagement with commercial MPS developers. We do this via 3 work streams. We have a regulatory subgroup, technology expo, and end user and education. Our end user engagement subgroup has done quarterly workshops last year and quarterly submissions to Altex, as well as posters at various conferences. And this year we'll continue with those webinars, this one being the first of 4. We have posters at various conferences again, and we'll continue to submit to Altex Corners. You can see all of our past webinars on our events tab. You can go to the MPS webinar series and see all of our past workshops and webinars in a convenient playlist. Our regulatory subgroup has been working on their DILI project, and their ISTAN letter of intent was accepted. They're working on a process paper and results paper. We launched a benchmarking survey on the use of MPS in regulatory. Megan had an invited talk, and we will do workshops at the 3D Tissue Model Summit and posters at various conferences. So we have an MPS Tech Hub on our website where you can look for relevant commercial tech providers, and you can filter by various tissues, diseases, and species. And we also have a publication hub with over 350 MPS publications with filters and search features. For our upcoming webinar schedule, more will be in May, September, and December. And then for this webinar, if you could kindly put all your questions in the Q&A tab, we'll do a panel Q&A with all of the speakers at the end. You can go to our website to learn more, and my email is on the screen if you ever are wanting to join our MPS initiative or become one of our sponsors.
Hello, my name is Richard, and I'd like to introduce the IQ MPS, which is the Microphysiological Systems affiliate. We are a collaboration of pharmaceutical and biotechnologies companies created as an affiliate within the International Consortium for Innovation and Quality in Pharmaceutical Development. The website is iqconsortium.org, and this provides a venue for appropriate cross-pharma collaboration and data sharing to facilitate industry implementation and qualification of MPS models. There are 4 aims for the IQ MPS. First is external partnerships and collaborations, which we try to do to help enhance the inclusion of industry priorities. Second being thought leadership, where we aim to serve as a thought leader for both MPS developers and stakeholder organizations in the industry implementation and qualification of MPS models. Third is cross-pharma collaboration and data sharing. And last is focused engagement between industry and regulatory agencies. There are 28 member companies for the IQ, MPS with over 150 active participants. As you can see, the member companies are in this list for this year. And this represents a diverse group spanning from drug safety, 3Rs, ADME, and PK/PD. This year's executive committee is comprised of Rhiannon David from AstraZeneca, who is the past chair, Kim Homan from Genentech, who is the current chair, Phil Hewitt from Merck, who is the vice chair, and lastly, James Gossett from Pfizer, who is the vice chair elect. And this executive committee provides leadership and guidance in the management of IQ-MPS business and other affairs, and it also implements strategic plans recommended by the steering committee. The steering committee is comprised of two individuals from each member company. They make the primary decision making, but they are the primary decision making body for the IQ-MPS. They establish the objectives, policies, and plans and actions that provide strategic oversight of the portfolio. And underneath that, there are four work streams, ranging from regulatory engagement, organotypic context and use benchmarking, etc. I will go into a little bit more detail in the following slides. And then lastly, it's the sub-teams, which have a variety of different roles, such as reviewer training, specific organs that they're looking at, and others. For the regulatory engagement workstream, the aim is to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS in an industry setting. Some of the strategic objectives include aligning on common understanding of performance standards and best practices for the qualification of MPS, building regulatory confidence in MPS, and facilitating harmonization across regulatory bodies, as well as encouraging the development of animal MPS together with human MPS. As for the organotypic manuscripts, the goal here is to serve as a thought leader for both MPS developers and stakeholder organizations in the industry for the implementation and qualification of these models. And each manuscript provides an overview of the available models, the cell types, important model features, relevant COUs, and test compounds for qualifications. As you can see from the list, the topics range from lung models to disease models to ocular models. Next is the pilot project work stream, where the goal is to provide a venue for appropriate cross-pharma collaborations and data sharing to facilitate industry implementation and qualification of MPS models. There are two workflows, the RFI, which is the request for information, as well as the RFP, which is a request for proposals. And we work with external entities such as Altis or PSTC to do this. Next is the Strategic Partnership and Communications Workstream, or SPAC. And the aim here is to develop external partnerships and collaborations to help enhance the inclusion of industry priorities. And the goal here is to develop as thought leaders for MPS developers and other stakeholders in the industry through internal communications, education, and supporting MPS adoption among the next generation of scientists. And we work closely with external collaborators such as 3Rs and CATS. So those are the slides I'd like to share. And if there's any questions, please feel free to email us at the link you see on the left or visit us at our website or LinkedIn page. Thank you.
Thanks, Richard.
Many thanks for the introduction. And thank you for the 3Rs Collaborative for the opportunity to present today. Today, I'm going to talk about the human iPSC cardiomyocyte proarrhythmia assay and its growing use in cardiac safety testing. Many people still think of NAMs as exploratory models, interesting science, but years away from real decision making. What I want to show you today is that perception is outdated. Some NAMs have moved from concept to reality, and that shift is already affecting how safety decisions are being made. So before we go any further, I need to define the term NAM. So NAM stands for New Approach Methodology, but you'll also see sometimes use non-animal methods. I think this definition on the slide from the US Environmental Protection Agency is a useful one. So NAMs are defined as any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment to avoid the use of vertebrate animal testing. Examples of NAMs are in vitro tests or assays which use human or animal cells, in chemicoassays and in silico algorithms. So if NAMs are no longer a future concept, the next logical question is, why is it happening now? The shift isn't being driven by a single regulation or a single breakthrough. It's the result of multiple forces converging at the same time. On the push side, drug discovery is inefficient, late-stage failures are costly, and animal models don't reliably predict human outcomes. On the pull side, human-relevant in vitro models are now demonstrating real predictive value. Together, these forces create acceleration, and that's what defines an inflection point. It's also important to understand that this didn't happen overnight. Regulatory engagement with NAMs has been building quietly for more than a decade through initiatives like the HESI-CIPR initiative and other FDA-led validation studies. The FDA Modernization Act in 2022 didn't start the shift. It just acknowledged that the science had matured, and since then we've seen the NIH, EPA, and other international agencies move decisively in the same direction. So let's unpack the last part. In December 2022, the FDA Modernization Act 2.0 was enacted into US law, marking a major change in how the FDA evaluates drugs before human clinical trials. This removed the mandatory requirement for animal testing for drugs and biologics and authorized modern non-animal testing methods. The Act explicitly allows the FDA to accept NAMs as evidence of safety and effectiveness. It gave the FDA and drug developers regulatory flexibility to use the most scientifically appropriate evidence. There are several misconceptions worth clearing up. Regulators didn't lower the bar, animal testing wasn't banned, and standards didn't change. What changed is how evidence can be generated and which tools are considered acceptable. Regulators are giving industry flexibility, but only where the science supports it. Which brings us back to the central question: which platforms are ready for adoption at scale? An adoptable NAM platform has to be predictive, reproducible, scalable, and defensible internally and externally. Without those characteristics, adoption stalls. And this explains why NAM adoption hasn't been faster. The hesitation wasn't philosophical, it was practical. Inconsistent data, lack of standards, throughput challenges, and regulatory uncertainty made leaders cautious, and rightly so. What's changed is platform maturity. We now have FDA and EPA tested assays, shared data sets, standardized workflows, and broad industry adoption. For leaders in industry, this isn't just a scientific shift, it's about avoiding late-stage failures, reducing unnecessary animal work, and making earlier, more confident decisions. So the natural question becomes, what does a validated NAM actually look like in practice? Here's an example of a NAM that's already crossed the line from experimental to operational. Cardiac safety assessment is an area where the cost of getting it wrong is high and where regulators have been deeply involved in defining what better prediction should look like. Since 1989, 14 drugs have been withdrawn worldwide due to dosage de points, TDP, a rare but potentially fatal ventricular arrhythmia that can lead to sudden cardiac death. It's characterized by a rapid heart rhythm originating from the ventricles, QRS complexes that change shape and appear to twist around the ECG baseline, and an underlying long QT interval. To mitigate this risk of developing potential proarrhythmic drugs, the International Council of Harmonization, ICH, issued two guidelines in 2005, S7B and E14. E14 is the ICH clinical guideline that explains how to evaluate whether a drug prolongs the QTC interval and increases proarrhythmic risk in humans. Its counterpart, S7B, is a non-clinical safety pharmacology guideline. In plain terms, it tells drug developers how to assess whether a new drug might delay cardiac repolarization and prolong the QT interval, which could increase the risk of dangerous heart rhythm disturbances such as TDP. S7B's objective is to identify a drug's potential to cause delayed ventricular repolarization early in development using preclinical studies, so the clinical risk can be better managed. S7B recommended evaluating the risk through two tests. One, an in vitro assay of IKR-hERG potassium block. IKR inhibition is strongly associated with the QT prolongation. Two, an in vitro QT prolongation study in animals. typically in beagle dogs. These measures have reduced the likelihood of drugs with hidden TDP liability reaching the market. However, these tests focus on surrogates, QT prolongation and hERG block, not the mechanism that actually causes arrhythmia. And so there are concerns that overemphasis on these endpoints may have hindered safe, effective drugs reaching the market. Human-induced pluripotent stem cell-derived cardiomyocytes have been proposed as a better model for predicting proarrhythmia because they capture human-relevant integrated cardiac electrophysiology in ways that traditional assays cannot. The argument is not that they replace the established models, but they address key blind spots in the older approaches. Human iPSC-derived cardiomyocytes express multiple interacting ion channels simultaneously and possess many of the structural and functional aspects of cardiomyocyte cell biology, thereby providing a system-based model for a system-based problem. Because arrhythmias arise from disturbed electrical conduction in the heart, Axion's Maestro multi-well MEA system is an ideal platform to pair with human iPSC-derived cardiomyocytes for assessing proarrhythmic risk. In the human iPSC cardiomyocyte MEA proarrhythmia assay, the cardiomyocytes are cultured on plates containing microelectrodes, allowing non-invasive recording of electrical activity following drug exposure. The assay measures changes in field potential duration, FPD, a QT-like surrogate, beat rate, conduction, and rhythm. Together, these measurements provide a direct functional readout of cardiac electrophysiology and key endpoints relevant to proarrhythmia risk. The Comprehensive In Vitro Proarrhythmia Assay Initiative, also known as CIPA, was a global FDA-led public-private initiative launched in 2013 to improve prediction of drug-induced TDP by moving beyond the traditional hERG and QT prolongation paradigm. To do this, CIPA proposed a mechanistic integrated framework with 4 pillars: multi-ion channel pharmacology with human cardiac ion channels, in silico human ventricular models, early clinical ECG analysis, and human iPSC-derived cardiomyocytes to verify integrated cellular effects. The human iPSC-derived cardiomyocyte arm was designed as a biological reality check to confirm whether predicted risks manifest as arrhythmia-like behavior in human cells for the in vitro cardiomyocyte pillar. It is therefore unsurprising that the MEA assay was selected as a platform of choice, as it offers a direct and integrated readout of cardiomyocyte electrophysiology. 2 multi-center CIPA studies involving pharmaceutical companies such as BMS, Eisai, Genentech, and J&J confirm that the human iPSC cardiomyocyte MEA assay can detect delayed repolarization and arrhythmia-like events linked to TDP. These CIPA findings directly inform the ICH E14 S7B question and answers that were published in 2022, which recognized human iPSC-derived cardiomyocyte data as supportive evidence for proarrhythmic risk evaluation. So what happened next? Let's look at how these preclinical validation studies have impacted IND submissions. An investigational new drug, IND, is a regulatory application submitted to the US FDA that allows a sponsor to begin clinical trials in humans with a new drug or biologic that has not yet been approved. A recent study by authors at the FDA, which is currently under review at the British Journal of Pharmacology, highlighted that between 2012 and 2023, 22 IND submissions were submitted to the FDA CDER with human iPSC-derived cardiomyocyte studies on MEA and 9 with voltage-sensitive dyes. These iPSC cardiomyocyte studies demonstrated higher overall accuracy in predicting clinical QT prolongation risk compared to standard hERG and in vivo non-rodent QT studies alone. Notably, to achieve comparable accuracy to the human iPSC cardiomyocytes, additional animal studies beyond the standard in vivo QT assessment were needed. Interestingly for us, the authors from the FDA went on to propose several scenarios where human iPSC cardiomyocyte studies could be considered as a replacement for in vivo non-rodent QT studies, shown here in this table. Furthermore, Now more than 12 contract research organizations offer this SIPR-style human iPSC cardiomyocyte MEA assay as a service using Axion's Maestro Pro MEA system. Just to remind you, human iPSC cardiomyocyte MEA data is not mandatory, but it's being voluntarily included in IND submissions, and CROs are offering it as a standard service. No one is forced to include this data, which tells me it must be delivering value. The FDA now offers a qualification pathway for NAMs. In 2020, the FDA launched the Innovative Science and Technology Approaches for New Drugs ISTAM program. This is an FDA program that supports the development of novel drug development tools, such as microphysiological systems, also known as organs on a chip, artificial intelligence-based algorithms, and other innovative technologies not covered by existing qualification programs. The program's goal is to qualify these new technologies to facilitate drug development and streamline the review process, ultimately helping to bring safe and effective new medications to patients faster. Axion submitted a letter of intent to the FDA in September 2025 for the human iPSC cardiomyocyte MEA assay to be accepted onto the iStand NAMS qualification program. I'm pleased to say that our submission was accepted onto the iStand program in December. We'll obviously keep you updated with the progress through the qualification process. A fair concern often comes up here: are we moving too fast? The key, I think, is context of use. NAMs aren't meant to recreate the whole animal. They're designed to have a specific system level complexity to answer a specific question. Better science drives better decisions and fewer late stage failures. So many thanks for your attention today. I hope you found this webinar useful. If we have time, I'm happy to take any questions now. Alternatively, please feel free to email me and we can continue this discussion. Thanks.
Thank you. Thank you all for attending. Good morning, good very early morning from Seattle. My name is Nick Geisey. I'm the CEO of Curi Bio. Today, I'm going to talk to you about some applications of our technology that we've been really focusing on the past year or so with the 3D model systems that we use. Mike gave a really great talk, and he covered a lot of the really interesting things happening in our industry. I've been doing cell and tissue engineering since back when I had hair, back in probably 26 years ago now. And it's been a really interesting ride. Early on, we would do kind of these types of experiments where we'd show people cells in weird shapes and weird situations, and they'd say, this is never going to be useful. And okay, yeah, maybe you're understanding some fundamental stuff, but really think about what you're doing this for. And then going to today, kind of seeing some of the stuff that Mike was highlighting, with iStand, with the work that FDA and other regulators are doing to accept NAMs. It's pretty exciting. There's certainly a lot of limitations that we have. Obviously, if you read the press, there's a lot of people saying you're never going to replace animals and you guys are over-claiming stuff. And I'm always very cognizant of, how far we've come and how much further we need to go. So I really do appreciate the 3RC organizing these types of committees to kind of really hone in on the kinds of problems and questions that our technology can answer today. And how we can be useful to folks. And what I love about this community is that everyone is super collaborative, and there's a lot of problems that have yet to be solved. And the folks on the other side of the table, the drug developers and who are probably listening to this, are always very kind of willing to say, hey, let's work together to make a new model. And that's where a lot of this technology comes from. So some of the applications I'll talk to you today have been done in collaboration with some of our partners in the industry, and so it's been very gratifying to see this. Anyway, enough of the preamble. So Curi Bio, we really focus on thinking about just delivering data to customers and delivering the kind of data that can drive a business decision. I'm a pharmacologist. I've been spending most of my career with engineers and physicists and being kind of the odd duck and not really understanding a lot of the underlying technologies. And I found that, when we try to combine these different technologies, engineering, physics, biology, it can get really messy. So at Curi, we really think about the problem as cells, systems, and data. And what I mean by that is at Curi, we do try to bring a holistic platform to the customer. So we can provide the cells and the disease models. We make those. We provide and we provide the devices that actually form those cells into kind of 3D structures or some kind of biologically relevant structure. We provide the machines that make measurements from those structures based on a drug interaction. And then we provide the analysis routines and the data routines that will actually derive meaning and useful information out of those measurements to drive that business decision. Now, we're not like holding a hard line that you have to use our cells, you have to use our machine, you have to use our data. You can use 1, 2, or 3 of these. We don't really care as long as you get the data that you need. But in some cases, it really helps to kind of reduce those pain points for folks and provide all 3 layers. I'm going to focus a little bit differently from what Mike was talking about and looking at 3D. Because in certain cases, you need to kind of add the complexity of 3D. Not every case, but in certain cases you do. So for a long time, we've been looking at skeletal muscle, cardiac muscle, and I've been doing this for a very long time. And one of the issues that we tend to have is that the cells don't last super long on these 2D systems, and so that kind of limits some different questions that you can't ask here. These are some data I performed with Tenaya. I've spent a long time trying to mess with 2D cultures and trying to get them to last longer. And on the top right, basically the point of the slide is to show you the top right, where we look at a control line in red and trying to look at the differences between that and 3 different disease lines. And you don't really see a difference in 2D. But once we actually start to get to 3D, what these folks showed is there you can start to see the disease phenotype and start to exacerbate the different phenotypes that you want to see in these systems. And what I also like about 3D is that we can kind of start to replicate some of the clinical markers that we see. So this is kind of a little bit of a gross slide. My apologies, especially this early in the morning. But these are some common experiments that folks do. On the top is a mouse anesthetized, where they have one of the muscles kind of on a force transducer. And you do force frequency curves. And on the top, really, I want you to focus on there's kind of 2 superimposed data lines. The top line is the healthy mouse. The bottom line is the DMD, or Duchenne muscular dystrophy disease, muscle disease line that shows reduced contractility. We could do this in mice. This is done in dogs a lot. Sometimes with the human EMG, you can do this in the clinic to help diagnose some of these diseases. And we're really trying to do that, again, replicating this in a dish, using the types of phenotypes, using the types of markers that folks in the clinic are accustomed to, just so that we don't have to kind of do a radical shift in the way that they're thinking about looking at these data. Much like the Axion data, where you can see a lot of that stuff looks like an EKG, and you can very easily kind of translate from what you see in the clinic to what you see in the dish. And again, here on our EMTs, you can see we have healthy cells in the dark green showing nice contractility, and the disease cells in dark blue showing not so healthy contractility. And Mike really honed in on scalability and usability and throughput and reproducibility. We sing this from the same choir. We think about our technology in a multi-well format. So this is just a really quick view of 24 tissues in a tissue plate. You stick it on the machine, you push a button, you don't have to do any kind of complicated math. You end up getting, hopefully, 24 tissues. Our typical success rates with the new users, like 95 to 100% casting success on the first try, with about a day and a half of training maybe. And then from there, you place it into the machine, you get signals that look like this, so continuous contractility in a non-destructive format, and then we can read out each one of those tissues, and then we can change that into a dose response curve to whatever we're looking at. And again, as I mentioned, we kind of don't care what cells you use. You can use ours or not, but we think about this as a sandbox. You can have a lot of biological inputs, so I just showed you some skeletal muscle, the rest of this talk will be cardiac. You can put those into our system. But one of the other benefits that you get is you get a piece of tissue, and you can take the culture media out during the experiment and do biomarker analysis, or you can do endpoint analyses too, and really kind of do a lot of multimodal experiments on the back of the tissue that you made. This is probably the only salesy slide I'll show you. Really, we think about a lot of different models. So again, we're going to focus on cardiac today, obviously. But we also do a lot of musculoskeletal, neuromuscular, and diabetic modeling of all things, because there's a lot of a big muscle phenotype in diabetes these days and GLP-1 therapies. So when we think about quality control and how good the tissues are, we really kind of think about phenotyping triage. And you know, a lot of things we look at are baseline contractile behavior. We look at responses to calcium because that's really important. I'll show an example of that later. Of course, pharmacological response is really where the rubber meets the road. Are these tissues going to respond the way that you expect when you add a drug? And also a really also important part, I'm a calcium imager by heart, is really understanding calcium and voltage kinetics. We have another system that measures stuff other than contractilities, measures fluorescence. You can see there's a bunch of tissues beating at the same time. I won't be talking too much about that. And then, of course, a really big hallmark is a force frequency response. First, talking about consistency, I showed you that kind of plate. There's like a hand holding a plate. This may sound silly to those who haven't done this before, but it took us a good year to develop. And the reason it did is because we really focused on consistency. Here we can see things like multiple healthy iPSC donors on the left. The top line is basically force. The bottom line is frequency of these same data. What I really want you to focus on is the middle and the right column, where we look at multiple differentiations from the same donor. We can see fairly good beat consistency across a month of experiments. We sometimes take these tissues out for a year, but typically most of the data I'll show you are like about a month. And then even the same donor across multiple experiments, we can actually see very good consistency across tissues and across experiments. We can start to compare something we get today in, I guess, the end of March versus something we got last August. Really, it's important to focus in on maturity. There's a lot of, you ask 10 people what maturity is, you probably get 15 answers. But in this case, we're using force frequency as the benchmark. And what we can do is do an electrical pacing routine. The machine has like a built-in electrical stimulator. On the left here, you could see without maturation, as we start to increase the stimulation frequency, there's no real response. But after a bit of maturation with some electrical stimulation, in this case, again, you could do whatever kind of maturation you want. You can actually start to see that nice force frequency response where right at about 2.5 hertz, you can see a better, stronger contractility. And we also see good relaxation kinetics too, because we're getting that nice peak that we're measuring from the contractility. We can measure a lot of different factors out of that. So I realize I'm talking way too much about the background stuff, so let's get to some data. Safety pharmacology is kind of where we started out, really trying to work with the SIPA initiative and find better ways to do safety. And of course, we always try to do the benchmarks too. So if you think of digoxin, for example, on the left, we can actually show how after we expose these tissues to digoxins, a couple of different lines. So we have like in-house cardiomyocytes and some commercially available lines. We can observe a rapid and pronounced drop in twitch force in really just a few hours. And comparing to the negative control, there's no effect. That's an acute response. We can also do chronic responses too. This is a 35-day experiment. I know this may be kind of hard to see, but long story short, those kind of gold bars are where we hit it with boli, would you say, I guess, of doxorubicin. And the lighter doses, you see most of the tissues they're okay, but some of the heavy doses, we have one tissue that just at the heaviest dose just dies right away. That first bolus kills them all. The penultimate concentrated dose is interesting because the tissues really get hurt at that first dose. They kind of recover over 2 weeks, and then you hit them again, and then that kind of is the kill shot, I guess you could say. Is they end up dying at the end of that. So we can start to see dose discrimination and different outcomes. Safety is, of course, very important, but we also think a lot about discovery. I showed you the skeletal muscle model. That really was our entree into gene therapy, and we're starting to do a lot more gene therapy on the cardiac side. And what we can see is experiments like this, where across these 24 tissues, we can, hopefully you can see my pointer maybe, you can see different concentrations of an AAV. And this is just a fluorescent marker just to show whether the thing got in there. And at the lighter doses, you don't really see much fluorescence. At the higher doses, you see much more fluorescence. And we can do this across different capsids. Here's a plate map just kind of showing you what to do. So we can start to really understand, okay, what are the different doses and responses and things like that. I'll show more data on that. We also have a lot of customers doing disease modeling. So for example, NYBPC3, it's a hypertrophic cardiomyopathy. Basically, your heart is super duper strong, caused by some mutations in some of the sarcomere proteins that make up your body. Again, you can use primary lines, you can use animal lines, whatever you want. In this case, we're using iPSC lines. We have a disease line that we're now making at Curi Bio that captures this disease. And then we also have a corrected version of that line, so we could actually start to do isogenic controls as well. And that's pretty interesting because when you start to look at these guys side by side, you see really clear differences. Right here in the light green, these are the disease line. In the blue is the corrected line. You see that, as you would expect, the disease line is stronger than the control line. Similarly, we have a heightened inotropic response to calcium. They're like super responders to calcium. But the real scary part comes when you start stress testing these. Kind of in the clinic, they'll make the patients do like a 6-minute walk test. And here we're just like really pacing these guys to make them work. So, you know, low calcium, the mutant tissues, they just can't follow pacing rates above 2 Hz. They just crash out. But as you increase calcium, the corrected line really continues to follow the pacing and the mutant line still struggles. And that really kind of shows you that they have a differential response that's clearly seen in the data. But you can also rescue them too. So here you can start to add things like mavacamten, which is a myosin inhibitor. Basically, dark blue is healthy, light green is too strong. That's the sick tissue, but you can actually knock that down with the drug. Similarly, you can start to attenuate the responses here with different drugs. I realize I've got like a minute left, so just crank through the rest. In the clinic, we also see these tissues get very fibrotic. There's a lot of like basically junk inside of the muscle tissues. And we were kind of, normally you don't like to see what's here on the left, but in this case, when Aisha saw these, she was very excited because this is exactly what you want to see in the disease line. We start to see these kind of little fibrotic lesions forming at the ends of the tissues, which we believed were indicative of the disease phenotype. And certainly when we actually go in there and slice and dice the tissues and stain them, we start to see alpha smooth muscle actin and TGF-beta1, which are markers of these types of disease. The last example I'll give very quickly is, rapid disease modeling. Sometimes you don't really have the time to make an iPSC line, or you don't want to go through the expense. You just want to kind of do a quick and dirty experiment. In this case, we wanted to see if we could take a healthy disease line and use siRNA to kind of model a disease. In this case, arrhythmogenic right ventricular cardiac myopathy. So in this case, again, we're taking a healthy line, hitting it with an siRNA to say, hey, can we make this disease? And yes, certainly we can. If on the left, we see the controls, basically the healthy lines. When we hit the siRNA, we see a higher variation in twitch force and a higher variation in twitch period. Really what those look like here. These are the actual data is on the vehicle. We have spontaneous and paced tissues. Look very nice, very regular. That's what you want. Negative control, yeah, very nice. That's a healthy line. But once you start to add this to add the disease model, you can start to see the variations in twitch force noted by the little peaks here, but also by the variations in rhythmicity as well. And so in this case, we can now start to either use an iPSC line or use a patient line or make our own little quick disease line and then use that to get those nice dose response curves. Again, our whole goal is to make this super duper easy, whether folks send us the compounds to do in our lab or we send them the machine and they do their own cells or we send them the cells. We really aim to make this easy. And even I, with my super, super stale pipette skills, they don't really allow me in the lab anymore, can get these types of experiments done pretty rapidly within a few days. So I hope this kind of give you an idea of where we come from and how we think about these problems. We're Curi Bio, again, based in beautiful Seattle. We're starting to get into the sunny season. So if you are in Seattle, please do give us a ring. Our building is right there in the middle, right on the water. So we can see some orca whales too in there while you're visiting. So thank you very much. Appreciate your time.
Thanks for the nice introduction and thanks for the opportunity of being here presenting BiomimX. So BiomimX is an Italian SME. We are a spin-off from Politecnico di Milano, and we are located in MIND, the Milano Innovation District, where private and public companies are located and works within the healthcare and biotech field. And our vision is to provide the pharma company and biotech with innovative tools that can allow them to discover the therapies of tomorrow by relying and catching human data that are already available today. And we do that thanks to our solution, which is called Beating Organs on Chip. That's a novel approach that combines microfluidics, human cells, and physical stimulation provided to three-dimensional models to offer clinically relevant in vitro models. In particular, we start using human cells. They can be cell line, primary cells, or iPSCs from patient, and we can inoculate into our UBIT-based device. And thanks to our patented technology, UBIT, we can provide reliable, tunable, and versatile mechanical stimulation to the cells that are cultured inside this device in 3D. And in particular, we can tailor-make the mechanical stimulation either to develop physiologically relevant models of organs that has a superior level of differentiation and maturation, or we can also use the mechanical stimulation to trigger pathological traits in the model, so to replicate complex disease in vitro by modulating the mechanical three-dimensional environment. We already demonstrated that our approach can be used in different models of organs and pathology. And for today's presentation, I will focus on our cardiac physiological model. Our cardiac physiological model is called Heart, and it's formed by 75% of human-induced pluripotent stem cell-derived cardiomyocytes and 25% of human adult fibroblasts that act as supportive cells. We embed cells into fibrin gel and inoculate into our platform. And after 7 days of mechanical stimulation that mimic the heartbeat, so it's a cycle mechanical stimulation provided at 1 Hertz, we can derive functional microtissue. In particular, as you can appreciate from the immunofluorescence staining, you can see that the cells has an enhanced three-dimensional organization, fibroblasts and cardiomyocytes interconnect, and cardiomyocytes retain the typical striation of the sarcomere. And thanks to the mechanical training we provide to the cells, we can improve the contractility performance. In particular, we can synchronize the cells and so achieve a synchronously beating microtissue that beats really as a syncytium. While in static conditions, cells still maintain the capability of twitching, but not altogether. And this is really important for functionality. And of course, we could detect that our model has a lower excitation threshold when mechanically trained and show a higher maximum capture rate. We use this model for cardiac safety, and I would like to thank both Mike and Nick to have the great introduction about the importance of having a better model in cardiac safety. And I will focus today on 2 contexts of use. The first one is detecting the QT prolongation and arrhythmic events caused by drugs. And the second one is for detecting contractility liabilities. In particular, we use Heart model in the first context of use, also thanks to another patented technology that we have in-house that allows us to insert electrodes in specific position in contact with the cells. And so we can be able to extract the electrophysiological parameter, and more specifically, the field potential for the entire microtissue. We can detect relevant endpoints such as beating period, field potential duration, field potential amplitude, and detect arrhythmic events or if the drug causes a beating cessation in the microtissue. We perform the pharmacological studies in this first context of use together with Accelera, which is the CRO in Italy, and we selected 11 compounds that are listed in the CIPA initiative that can act on single or multiple ion channel. And in particular, we develop SOPs that are compliant to the ICH F7B Q&A guideline to perform the test, and the preparation and the assay are really standardized. We also set a quality control for the development of the microtissue, so only Heart that show a coefficient of variation of the beating period at the end of the culture below a certain threshold that here was fixed at 25% were used for the pharmacological campaign. We inoculate the drugs in an incremental manner, and we compare the concentration of drugs found effective in heart with the Cmax or free effective therapeutic plasma concentration. So to calculate safety margin and also having an integrated risk assessment. We started by using training set compounds such as the fetylide, nifedipine, and mexidecine, which are known to selectively block specific and single ion channel. And as expected in the case of the fetylide, we could see that we had an increase with respect to the baseline of the field potential duration corrected with the Friedrich methods. For the nifedipine, we could detect a decrease of the parameter, while for mexidecine, we didn't detect any specific trend. But when we looked at the spike amplitude, we could see that mexidecine was the only drug causing a statistically significant decrease in the amplitude, and this was coherent with its effect on the sodium channel. We then repeated the test for all the set compound acting also on multiple ion channel. Here I'm just reporting as an example the quinidine, and we could see again that as expected that the drug caused a field potential duration prolongation in a dose-dependent manner. We could also detect arrhythmic events at two concentrations, 0.1 and 30 micromolar. And we then also define a specific threshold to correlate the concentration found effective in heart with the Cmax of the patient. By repeating the screening on all the compounds that I mentioned before, we could also set the predictive performance metrics of heart that was found to have a sensitivity of 83%, a specificity of 100%, and an accuracy of 91% to detect QT prolongation, and similar sensitivity, specificity, and accuracy to detect arrhythmic events. Moreover, following a more integrated approach, so merging data both from QT prolongation and arrhythmic events, we could categorize different classes of compounds to have a high risk or a low risk to the onset of the TDP. And this categorization matched the FDA label. So UHeart was a preclinical tool able to detect QT prolongation and proarrhythmic effects in vitro, and can be offered as a predictive alternative to more standard assays such as HR and in vivo telemetry studies in animal. The second context of use is the contractility assay, and in this case, we can analyze video of spontaneously beating microtissue, and the relevant endpoint are the beating period, the contraction amplitude and contraction velocity, which works as a surrogate of a force of contraction, relaxation time, and contraction duration. In this case, the pharmacological campaign was performed together with the top 10 pharma, and we select both approved and tool compounds. And the compounds act either on a specific ion channel as before, or can be defined as a modulator of the structure of the cardiomyocytes and has different mode of action. And also here we correlate the data with the Cmax or IC50 in vitro. I'm reporting here as an example the positive control isoprotenerol, and in fact, as expected, we could see that UHeart found it as a positive inotrope, positive chronotrope, and positive lusitrope. And we repeat the test for all the other categories of drug, like sodium channel modulators, calcium homeostasis modulator, and sarcomeric activity modulator. And we could compute again the predictive performance, showing a sensitivity of 63%, specificity of 80%, and accuracy of 67% to detect the inotropic effect. But what for us drive our attention was the fact that UHeart, so a three-dimensional mechanically trained model, could outperform to the assay, especially for the drugs that affect the calcium homeostasis and the sarcomere of the cardiomyocytes. So again, UHeart was able to provide detailed contractility insights into drug-induced cardiac changes, and can be the assay of choice for specific classes of compounds where more complex environment and complex structure are needed. Just to mention some future direction, we are also developing a chamber-specific model. We mechanically train either atrial and ventricular microtissue that retain the typical phenotypic characteristics, and we could also detect the field potential of the microtissue, and we also perform a proof of concept of drug screening to assess how this model reply to different kind of drugs. And we are also expanding into disease model, and in particular, we can use our technology to develop in vitro model of cardiac genetic pathologies that can be used for developing personalized therapies and for diagnosis. We are indeed involved in different consortia, and we are now generating the models from IPS cardiomyocytes that derive from the patient carrying the genetic disorder. And we also have a model of cardiac fibrosis, in particular our USCAR model that encompassing a population of fibroblasts was already used for drug discovery and efficacy screening, both with small molecules and gene therapy. And we are also expanding toward incorporating cells from the immune system thanks to a Marie Curie network miracle. Just to conclude, so we have capabilities to perform safety studies. We can provide services for testing cardiac safety, both of small molecules or new modalities on our qualified model. We can partnership with you, so we have also technical and biological expertise to develop and complicate at the level needed our model, tailor-made for your needs. And we can also provide our technological platform under evaluation and licensing to test the setup in-house. And with this, I would like to thank all my team and thank you for the nice attention. I will be happy to reply to any of your questions.
Hello, everybody. Good morning, good afternoon, or whatever time zone you are in. I hope you can hear me okay. So I would like to introduce our 3D cardiac microtissue model today. We have that set up both for cardiac toxicity or cardiac safety, but also for efficacy screens. And I have some examples for both of those opportunities. So as everyone knows, some 3D models may come with some challenges, especially if you are talking about self-assembly microtissues. They come with high variability, sometimes limited scalability and throughput, and high complexity for the end user. On the other hand, 3D models definitely offer some advantages as advanced cell maturation, more complex and holistic in vitro environment, integrating different cell types into one microtissue, which then leads to an integrated tissue response. At Ncardia, we have developed a cardiac 3D microtissue model in collaboration with our partners at the LUMC, the Leiden University Medical Center, where we combine 3 different cell types, which is iPSC-derived ventricular cardiomyocytes together with vascular endothelial cells and cardiac fibroblasts, and they are all differentiated from the same IPS line, so you have like an isogenic model. The way we are doing this is using our Tican robotic system to set up the microtissues. So we start with throwing the cells individually, and then they are pre-cultured for 3 days in their individual optimized media. And then at a given day, we dissociate them, and then we mix them together at a 70% cardiomyocyte and 15% each of the endothelial cells and cardiac fibroblast ratio. The formation of the microtissues is done in 384-well plates with the robotic system, which means that you can actually handle a lot of these plates. It also increases the homogeneity of the microtissues across the plate. We keep the cells in culture or the microtissues in culture for a period of 14 days before we use them for our actual readout. And the preferred readout for the 384-well plate format for a functional assay is a calcium transient assay. So what we usually do for an acute assessment of compounds is you load the cells with your calcium dye of choice, and then you incubate them for a while before you actually challenge them with the drugs within our reader, which is in this case the Hamamatsu FDSS device. So with this, you end up with having homogeneous microtissues with a low variability. It's in the 384-well format, so it allows for some decent throughput, and it's also easily scalable. And you have a functional assay that you can use both for acute measurements, but also for chronic assessment of compounds, where you just would have to revert the treatment so that the treatment comes first, and then after a given time period, which could be like 3 days or a week or whatever, you would apply the calcium dye on top of that. And then the only difference in the assessment of the data is that you would have to only do the vehicle control normalization instead of also running a baseline normalization. I can assure you that if you use the robotic system, it's really a relatively smooth and process that is also not requiring a lot of time. So formation of the microtissues, once you dissociate the cells, is really happening within minutes instead of hours. And as said, you have a very reliable and homogeneous microtissue population. So to show you some examples, initially we tested different cell numbers for the microtissues across different batches of cardiomyocytes. And as you can see here in the XML image from 384-well plate at day 7 after microtissue formation, you can see that you have microtissues in each and every well of the plate. So we have like 0 failure rate regarding the formation of the microtissues. When it comes to the cell size, we compared different batches of cardiomyocytes with different total seeding densities. So this is like the total number of cells of the microtissue. And then we did these measurements after 1 day, 7 days, and 2 weeks of culture. And as you can see, especially for the 10K and 5K microtissue size, there is really a low variance. And also over the period of the culture, there is only little shift in the size of the microtissues. Then next we were challenging them with just a couple of tool compounds. So in this case, what you can see is when you hit them with isoprenaline, you can see that you have like an increase in beat rate when compared to the time-matched vehicle control. But what we also could observe is that you also get like an increase in the calcium transient amplitude, which shows that you not only have like a chronotropic, but also like an inotropic effect. Then when you hit them with nifedipine, a calcium channel blocker, you can definitely see that there is like no change in the beat rate, but we do definitely see a decrease in the calcium transient amplitude, indicating that the compound is doing the job that it's supposed to do. And then on the other hand, if you hit them with the BK8644, which is a calcium channel activator, you can see that again, nearly no change in the beat rate, but we do see a significant increase in the amplitude of the calcium transient measurement. So with this set of tool compounds, we could at least confirm that in the calcium transient assay, all the two compounds that act directly on the calcium regulation are acting as expected. What we also did was to have preliminary data on a fibrosis model. So what is known is that TGF-beta1 is a key mediator of cardiac fibrosis, promoting extracellular matrix deposition and cardiomyocyte hypertrophy. And elevated NT-proBNP level correlate with heart failure, myocardial remodeling, and fibrosis. Also, it is known that endothelin-1 is a potent vasoconstrictor, and it contributes to cardiac remodeling by stimulating hypertrophy and fibrosis, and can synergistically amplify the effect of TGF-beta on mediating fibrotic and hypertrophic signaling pathways. So what we did was to hit our microtissues with some concentrations of TGF-beta, and then in response, we were measuring the release of NT-proBNP into the medium with a MSD assay, and as a positive control, we also used a single concentration of endothelin-1. And what you can very easily see here is that with you have like an underlying basic release of NT-proBNP into the medium, but then if you increase the concentration of TGF-beta, you can see also like an increase of the release of NT-proBNP. And as expected, endothelin-1 is a very strong inductor. And this is an assay that you can very easily add on top of the calcium transient measurement, because you can sample from the microtissues at any given time point to assess, for instance, these markers, but also any other biomarker that is supposed to be secreted into the surrounding medium. in our collaboration with the LUMC, we were also establishing an assay where you can actually look for compounds that will revert a certain disease phenotype. And the model of choice here was an IPS model of CPVT. And CPVT stands for catecholaminergic polymorphic ventricular tachycardia. This is a rare inherited disease that is caused by mutations in the ryanodine receptor genes, which results then in abnormal calcium handling. In patients, ventricular arrhythmias are usually triggered by stress or exercise, and flecainide is used therapeutically to suppress the abnormal calcium release and rescue the phenotype. So now when you do that in vitro, you can very easily see here on the right-hand side that with like no stress inductor, the calcium transient of the CPVT microtissues are looking more or less normal. However, when you induce the stress by adding certain triggers to the cell, you can very easily and nicely see that this is inducing arrhythmia. And then when you put the flecainide on top of that, it brings everything back to the like the normal phenotype. So now when you calculate an arrhythmia score from these microtissues, you can see that when you add the trigger, approximately 90% of the microtissues is showing a phenotype. And then on top of that, you add the flecainide, which then again brings approximately 90% of the microtissues back and rescues the phenotype. And this is something that you can't mimic when you're using only the cardiomyocytes in a 2D system. So this is here a very good example where it actually needs the 3D environment and the 3 cell types playing together to get these phenotype up and ready. And then you can use that to screen compounds. And that's what we did. So we tested 2,000 compounds from a drug repurposing library, which took a bit more than 2 days to run all of these 2,000 compounds. It was a fully automated workflow. So from microtissue generation to compound addition and the assay readout, it was all done with a robotic system. There was no loss of microtissues across the entire initial screen of 2,000 compound. And as mentioned, the compounds were screened in triplicates and completed within 2 days. Across the screen, 97 positive compounds were identified, which is a 40.6% hit rate. And the number of compounds, or let's say the therapeutic areas of the compound hits that were found is spread across a relatively wide type of compounds. We had like anti-cancer agents, compounds acting already on cardiomyocyte, but also compounds that are involved in immune response or neurological disorders. So from the quality perspective, the per plate QC confirmed expected response to the positive controls. We had less than 10% interval variability in the microtissue size across the whole screen, and the average prime factor was 0.6 across the entire screen. So the key outcomes of this exercise was that the LUMC was able to sufficiently differentiate enough of the 3 cell types from the disease cell line, as well as we could form the microtissues in-house here, so that we could support all the phases of the screen, which also initially included a run-in experiment. Then the screen identified compounds that were actually not previously evaluated in a CPVT1 model or in any clinical study. So that was very good for our clinical collaborators to come up with some hits from very interesting compounds that were not identified previously. And at the moment, as we speak, we are actually running another screen with another set of compounds, resourcing some compounds, and then also doing some potency tests on the hits that were identified initially. So then again, when you look at the hit rates that have been identified during this screen, there is definitely especially in the neurological therapeutic area, we had a lot of hits that were identified, but then also some anti-inflammatory compounds, and then compounds that are already identified as being active in cardiovascular diseases, but also some of the antiviral compounds were identified as positive hits, and that's perhaps nothing where you would initially look at when you think about this type of disease. And I think that was already my last slide. Yeah. So the key takeaways is that our 3D microtissue model, which is called Heart in a Box, is designed to be used in high throughput screening because we run that in 384 plates. The microtissues require a low number of cells and are very homogeneous with low variability. We can generate up to 1,500 data points per day based on a calcium transient assay as a functional readout. And the setup can be applied not only to wild-type iPSC cell types, but also to any type of iPSC-based cardiac disease model to study phenotype rescue. And the model is also suitable for the assessment of cardiac safety and especially also structural cardiotoxicity, combining not only the functional readout, but also some other endpoints. And with that, I would like to thank my colleagues here at Ncardia, but also the colleagues at the LUMC who did most of the work on that model.
Thanks so much. So we have time for a couple of questions, and we do already have some. So one of them is 3D microtissues only measure calcium transients, but not force production?
Yeah, that is correct. Our standard readout is the calcium transient assay, and that is mostly because of the throughput. So our goal was to have a system where you can assess really a lot of compounds in one run. So that was the goal here, and we do not assess any force at the moment.
Oh, thank you. Another one, what is the readout for these 2,000 compounds? Is it cell death?
No, that was actually the calcium transient. So it's a functional readout, and since we run the screen on the disease model, it was actually to look if the disease, like the arrhythmia induced by the trigger, can be reverted, similar to the positive control flecainide, which is already prescribed in the clinic. So the readout was the reversion of the arrhythmic phenotype in the CPVT model.
And one more, how is the initial cell population changing over time, especially endothelial cells? Did you just rely on the size change?
We did some initial immunostaining of the cell types just to make sure that all of the cell types are present. What you usually see is that you have this spheric microtissue, and you have the endothelial cells forming kind of a network in the 3D space, and then the cardiomyocytes and the fibroblasts are integrating into that. So just to make sure, so we do not add any extracellular matrix here. So it's a self-assembled microtissue. It takes approximately 24 to 48 hours to form microtissues that are already relatively solid. They will then grow slightly in size over time, but as I showed on one of the graphs, there's not much size increase. We did not quantify over time if there is any change in the original percentage of the different cell types, but if you look at some immunostainings, you would see that still the cardiomyocytes are the majority of the cells. And interestingly enough, we don't add any, let's say, cell proliferation suppressor. And initially, over the first 3 days of culture, the fibroblasts do proliferate, but they don't do that once you put them into the microtissue. So we do not see any outgrowth of fibroblasts or a super big increase in the size of the microtissues. So obviously there is no enhanced cell proliferation going on.
Great, thank you.
Well, thank you to 3Rs Collaborative for the opportunity and the great speakers that walked us through the technologies that they have so far. I am going to present about Exir Bio's platform, which solely focuses on non-human primate models. And the goal at Exir Bio is to develop non-human primate in vitro model, which includes stem cell-derived cells and tissue, including cardiomyocytes. So full disclosure about any cells that are used in our platform are sourced from known colonies of non-human primates that hold full genome sequencing of each animal that will help us in identification of mutations that correlate to disease, and also characterizations of these animals in terms of being born in captivity. So the agenda for my talk is about the use of non-human primate stem cells and derivation of various cell types from cynomolgus macaque and different non-human primate species, and how this addresses the industry challenges that we have these days about in vitro models and the benefit of non-human primate in vitro system for NAMs and applications in principles of 3Rs. So before I dive into the actual products, I would like to highlight some of the aspects why primate stem cells are more challenging compared to non-human primate stem cells are somewhat more challenging than the very well-established field of stem cell biology in human. So as you guys might know, this will be a little bit of scientific venture here that we take together, that in earlier stages of embryo, we have this clump of cells called inner cell mass that basically are the in vivo counterpart of the iPSCs that we are using. Any iPSCs that are used to generate cardiomyocytes basically is supposed to be morphologically and characteristically like the cells that are in day 5 of the early embryo development. And the first embryonic stem cells actually were derived from a macaque in the primate taxa in mid-90s. The foundation for the technologies that are used currently to derive cardiomyocytes or other cell types from stem cells, iPSCs in vitro, are based on the work that was done by Yamanaka in developing iPSCs in 2007, 2006, and 7, that skin cells or blood cells were reprogrammed to iPSCs that from which we can derive cardiomyocytes or other cell types almost similar to the embryonic features. Now the question is, could that be applied to non-human primate? How similar they are in terms of physiologic features, and could we replicate that easily for non-human primates? Like I said, that the challenge in verification and validation of of the stem cells is that if we inject these stem cells into an embryo, if the quality of these stem cells are good enough, they can mingle and integrate with the existing cells inside the embryo. And as you see in the case that when you inject the mouse stem cells into a rat embryo, you might form, if the quality is good, the quality of injecting stem cells are good, you can form cross-species chimera, and this is the examples that has been shown. And in some examples, you could even use a knockout mouse to generate organs inside animals. But these have been all done in experimental methods. But what about the humans, using human stem cells in other animals? A lot of researchers have been trying to replicate that to generate human organs in other animals. But the challenge is that the quality of the stem cells of primate taxa in general is not as good as rodents. So when we come to non-human primate model, we found that this has been more challenging because generation of the stem cells for non-human primates is not as straightforward as it is in human model and rodent model. So we were able to generate the high quality non-human primate stem cells, which is on feeder-free condition, and it is easy to differentiate, start working on these cells to differentiate into different models, including the cardiomyocyte model and the neural model. And here I'm showing that the examples of the cells that were generated from non-human primates. So we were able to validate these cells into in vitro system to see whether or not these cells are high quality to integrate into cross-species embryo. And by injecting these cells into in vitro embryo, we were able to validate whether the cells are able to integrate into the embryo and grow in vitro. And here are the cells that they built with the red fluorescence injected into the early embryo to be able to mingle and integrate in the near inner cell mass. In the next phase, we wanted to validate whether or not the non-human primate cells, how compatible they are with the human. We have started working, generating the mixed colonies of the cells from between human and primates and differentiate them to the cardiomyocytes to see whether or not they can form functioning layers of cardiomyocyte in the dish. And the applications of this is whether or not we can use these cardiomyocytes for preclinical testings of human drugs. A lot of the tests are being done on non-human primate models that include cynomolgus macaque, and a lot of cardiac toxicity are being done on cynomolgus macaque as animal model for humans. But the question is how comparable these cynomolgus macaque models are to human. Are they predictive or not? And using these methods of derivation of cardiomyocytes from non-human primate iPSCs, we are able to make a side-by-side comparison with human before we spend time and money on expanding or exploring these items in NHPs, specifically cynomolgus macaque in vivo. So now I'm going to show you a few examples of how we have been using the NHP stem cells for drug safety evaluation. As you know, for the current industry challenges is how we reduce the use of animals in drug safety screening. And mainly I want to highlight the FDA roadmap that was released in April last year, that specifically focusing on non-human primate use in preclinical safety. As you know, the challenge for non-human primate industry or for preclinical safety is that time to market for drug development is approximately 12 years for each compound, and it costs about $2 billion. And when we are at the stage that the drug fails before clinical trial in human, we are almost 6-7 years in, and we have already spent several $100 million. And the failure at this stage costs a lot for patients and for pharmaceutical industry that have spent years for development of the drug. So the goal at Exir Bio is if we can use the in vitro models of primate to catch this failure early on and basically fail fast at this stage using NHP in vitro models. Now, how we do that, as I mentioned, we bring the stem cells, high quality iPSC model from non-human primate model and differentiate them into various cell types, including cardiomyocytes and the neurons, and we try to screen the drugs early on cardiomyocytes or iPSCs of non-human primates. Another applications that we have seen in collaboration with pharmaceutical companies is that if the toxicity is observed in the NHPs, we can go back in vitro using NHP iPSC-derived cells to illuminate the mechanism of failure and retry the drug candidate back into animal model. So we mainly focus on 3 areas with access to the animals that from which we derive about 20,000 animals, and that we are trying to use blood samples from these animals that are picked in clinical routine to build a big biobank of stem cells and iPSC-derived cell types, including cardiomyocytes. And as I mentioned earlier in my talk, each of these cell types in the biobank have their genome sequenced. So now this biobank of stem cells and stem cell-derived cells can be a great resource that reduce reliance on live animals for drug testing. And the access to the genome for each animal helps us to be able to determine whether or not genome engineering and modification can be done. And I walk you quickly through 3 case studies. And one of them is in the space of protein degraders that have shown several toxicities that are not compatible between human and non-human primates. And this goes back in the drug category of protein degrader, the thalidomide, that a lot of people know the history that in 60s caused toxicity in the pregnant women, result of babies born with birth defect, and goes back to several transcription factors that were involved in that early embryonic development. And that was because the drug was tested in rabbit and rats, where unfortunately they couldn't detect the toxicity at the time. However, non-human primate was the perfect model to be able to detect the toxicity, including some of the transcription factors that are important in that aspect, mainly SAL4. So now the idea was, when we were approached by a pharma partner, whether or not we can use our stem cell models for both in vitro screening before investigating, spending time and money in doing in vivo in non-human primates. And we developing reporter cell lines that were used to differentiate to many cell types, including cardiomyocytes, to be able to detect the effect of drug on this protein degrader on the cells. So I'm going to quickly go. We used thalidomide as the positive control that showed degradation of the target protein. Degradation of target protein results in loss of this red fluorescent protein. As you see in the MSO control, we don't have degradation compared to 3 replicates of adding thalidomide. And the beauty was that when we ranked these 4 compounds from the pharma company before they go tested in monkeys in vivo. And as you see, 4 hours after different concentration of the drugs, 12 different concentration and 4 drug candidates, along with thalidomide and other positive controls and negative controls. And we were able to see 24-hour exact concentration that shows toxicity. When the loss of red fluorescent means toxicity or the negative side effect of the drug on the primate cells that were used for differentiation or culture. And as you see, this was replicated 3 times, and we had consistent result, being able to determine EC50 before even spending any animal or time or any non-human primate being involved in any study, to be determining the toxicity level of drug A, B, and C, and D, and exact EC50 concentration without any in vivo studies in non-human primates. So we repeated the same thing in human stem cells, so side by side between human and monkey, and we were able to find out after that, before even the drug is being tested in any animal, that by side comparison in vitro between NHP and human, that this protein degrader that was being evaluated in 4 drug candidate has different outcome in the cynomolgus macaque and human. And that in vitro result helped the pharmaceutical company decide that even non-human primate is not a good model for testing this drug, and exclude that and submit that as a weight of evidence to FDA. So in summary, that we have been showing that the reporter cell lines for protein degraders, this work is now being extended to cardiotoxicity of the same cell types and the compounds that have been seen in NHP versus human. The second and the third case studies that I cannot be quite relevant directly to cardiac toxicity are the gut organoid models that we have been using. And also the third one that I wanted to mention to you is the neural model, all of which have been used as weight of evidence and context of use in submissions to the FDA. And with that, I would like to summarize that in terms of comparative in vitro human and non-human primate models are the model of comparative aspects between the two species have been used to validate the mechanism of action. Is it conserved between the species before we get engaged into non-human primate in vivo? And if we see any toxicity in the animal, we could investigate that using NHP in vitro systems. And finally, the parallel in vitro in vivo studies could be done in NHPs that is impossible to be done in humans. And with that, I would like to conclude and thank the organizer for the conference, for this webinar, and happy to answer questions.
Thanks so much, Murtaza. All right, so we'll go ahead and do the panel Q&A. So if all the speakers could come back on camera, but leave yourself muted. I think we have a question that was from after Mike's talk. So can you elaborate a bit on the path from when the system was first used by CROs?
So that's a great question. CRO adoption was very, very early on. I'd say perhaps more than a decade. The system and the assays have been used in CROs. I think probably Cyprotex was one of the first companies to take that technology on, but I would say that was very early in the kind of life of the assay.
Great, thank you. And then I think this one was from after Silke's presentation. With ET1 application, do you also see increased cardiomyocyte microtissue size?
We didn't specifically look at that time. So I haven't done the experiments myself, but it's definitely something worthwhile to look at. So usually measuring a size increase in the cardiomyocytes by a hypertrophy inductor is not easy if you do that in a 2D model. So I would also set my hopes on a 3D model, but as said, we haven't specifically looked into that. Especially you would have perhaps more specifically image for an increase of the size of the cardiomyocyte in the 3D space. And honestly, I'm not sure if our imager would be equipped to do that in an adequate way, but it's definitely a good idea and also something that we already have planned to run in the future.
Great, thanks. And then this question for Murtaza, just to clarify, the first model is a tetragenicity model investigating the drug effect on differentiation, correct?
Yes, the protein degrader is teratogenicity. We are extending that. We know that. The model that I showed was teratogenicity and early differentiation of the cells into different lineages, ectoderm, mesoderm, and endoderm, and on some of the transcription factors. But we know that in the collaboration that the cardiac toxicity that was observed, some of these drugs show toxicity in human that were not caught in cynos.
Thanks. All right, general question for the whole panel. What are the biggest concerns that pharmaceutical companies raise when considering NAM products or services? Cost, throughput, predictive accuracy, functional relevance, something else?
I can quickly answer that. One of the challenges that we have been, I don't know, the camera, I tried to turn it on, but it's off.
That's okay.
The challenge was the validation of in vitro to in vivo. And we know that there is a lot of data from human safety that could be validated in vitro through reverse translation. We have the in vivo data, we replicate the data in vitro. But unfortunately, we don't have a lot of extreme safety cases in human patients, as we have in animal models. Because by the time the drug gets into clinical trial, already all those extreme safety evaluations have been done in animals. So it's true that we have some safety issues in human, but they are not extreme versions. And this is still a big challenge in the field, how to correlate in vitro to in vivo.
Thanks. anyone else want to comment on this one?
I would say from the examples that you gave, it's more or less a bit of everything. So it all factors in. So like with our model, throughput is not the issue. But then again, you also need something to compare it with. And is this now the already well-known 2D cardiomyocyte system like Mike described for the MEA? Or is it rather than really the in vivo data and then which species to look at? Because we are in the human space, and if you compare that to any animal model, how predictive is that? So it's a bit of everything.
Thanks. Anyone else?
I can just add a comment. I think the paper that I showed that was it's still in review, but I think it's a very exciting paper because it actually shows how the in vitro data compares to not only the existing tests but also the in vivo human data, and that those kind of data sets are quite rare. And then, once you have data sets like that, then you can start to answer those questions of where can the in vitro tests actually be useful. And sometimes it's easy to speculate, and I think some of the comments we heard in the talks, I think Nick mentioned it about some of the hesitation moving towards NAMS-based models is based on that idea of, are we moving too fast, etc. But until we have those data sets where we can see how the new tests compare to the existing tests, compared to the clinical trial data, it's quite hard to make those jumps. So my point being is the more of those studies, the better, I think, for the field.
Thanks. Anything else for this one?
Sorry, if I can just jump into this consideration, I agree with all the comments, and also I think now pharmaceutical companies are more aware of the capabilities and are also accepting that integrated methods can be the response to their needs. So not just one platform fits all the problem, but different one tailor-made on the context of use specifically and on their needs, basically. So I think this was something that in the latest year was really the game changer.
Thank you. All right, another one for everyone. They'd like to hear the panel's thoughts on use of these or other MPS technologies in academic research. I'm in Animal Oversight Research Administration at a university. I want to help our researchers stay up to date and generate results that will translate well to possible clinical assessment.
Is no one going to take
that one first? Yeah.
I'll go to break the silence. I think in the academic setting, you have a great advantage that you can move fast, right, and test lots of different new and novel technologies. I think where some of the challenges come in with that adoption then into industrial settings is that kind of burden of proof and that the larger data sets that are required to kind of validate. So I think in the academic setting, there's lots of kind of great emerging technologies. The challenge often is making that reproducible and scalable, that when it moves to kind of the industrial setting.
Thanks.
I think scalability is one aspect, and then also in academia you can do all the really fancy stuff and play around with all different kinds of models and technology and whatsoever. But then in the end, you want to have something that is more standardized, so that the end user in industry or any of the CROs providing this as a service all will have like similar results with this using the same platform.
Thanks. Anyone else want to comment on using MPS in academics? Okay, we have a couple questions for Silke. Please comment on if endothelial cells are necessary to conduct 3D microtissues.
I mean, I don't think that they are necessary to have a functional microtissue. So the cardiomyocytes will also just like start contracting and working with the addition of the fibroblast. It won't work without the fibroblast, or at least not for as long as with the fibroblast. We added the endothelial cells because that's the three major cell types that you have in the heart. So it's not only cardiomyocytes plus fibroblasts, but it's actually all the three. So it was really to have a more holistic model of the three major cell types of the heart.
Cool. Thanks. And do these 3D microtissues present both MYH6 and 7 or majority MYH7? Can you do the similar construct for skeletal myoblast?
We never did that with skeletal myoblast, and I should know the expression data, but I don't know them from the top of my head. We do have some RNA-seq data of the microtissues, so whoever asked the question, just like contact me directly. Happy to answer.
Great, thanks. Great. So if there's any other questions, please go ahead and put them in the Q&A. We'll give it a couple minutes, and then if there's nothing, we'll go ahead and end early. All right, I think we can go ahead and end a little bit early. Thank you so much for all of your talks and for all the questions. And yeah, I hope everyone has a great rest of your day. The recording will be available in a few days and everyone will be given an email to it. So thank you so much.
Great.
Thank you.
Thanks, everyone.
Bye.
Thanks for the event. Bye.